We're at the IAS with Frank Tomaka, who's clinical leader of the HIV program with Janssen, and we appreciate your taking the time to be with us. And actually, this is the second time because the first time didn't work out. Technical difficulties. So not only does it happen in science, it happens actually in our video programming too. But it's important to know that, that a little bit about you, and I think in our previous program we did talk about your background and how you got into the field, and that's a nice thing to hear, because we're all, here for different reasons. Some of them are similar, but we all have our own stories.
Well, thank you for the opportunity. It's really an honor for me. I've been very privileged in life. I was able to become a physician, which I think is a great privilege for anybody. And I had been a HIV specialist, first in New York and then in the Miami area for 15 years, which was an exceptional privilege. For people to let you into their lives when they're the most vulnerable.
Right.
But about 13 years ago, I joined a Johnson & Johnson company that was working on the most exciting pipeline of antiretrovirals at the time, Tevotec, and I got to work on darunavir or Persista and Intolince or Etravirine. And then 7 years ago, I was given the once-in-10 lifetime opportunity to work on their HIV vaccine program with really no experience as a vaccinologist, and it's a very different world than working on drugs or small molecules, but they obviously had some confidence in me, or they were possibly desperate, but 6 years later I'm still here, so I must be doing something right. And
it's hard to imagine that this took place in such what you would consider a short time.
yes.
All this work, all these drugs, and I know that the plan is always to be looking ahead 10 years. That's the vision that you have to have. But it's amazing that all this happened in 10 years.
Yeah. I think many of us never thought we would be this far in our lifetime. I'm sure many of your members and readers remember 1996 when the paradigm changed and people who were certain they were going to die were given a new life. But it was just the cocktail could have been 22 pills a day without other medications for prophylaxis. And for how many years did we as physicians and patients say, oh, if they could only make it so we could take one pill a day. And how many options are there now for one pill a day of medications that work better and are
really
safer?
a short span of time.
Really?
That's amazing, yeah.
And then no one thought we would, I don't think very many people thought we would ever see a vaccine in our lifetime. And although we don't have one yet, I, as well as many others, are very encouraged we will.
Well, I think that's that's the lead into the candidate you have going. It's time to talk about that.
So at Janssen Vaccines, we are developing a preventive HIV vaccine that is going to have global utility. So as most people know, there are a variety of HIV subtypes throughout the world, or what we call clades, and they're very different genetically, and therefore it's hard to for the immune system to mount responses towards all of them. They may respond to a clade C or attack a clade C virus, but not a clade B virus. So you would need vaccines for different vaccines for different parts of the world. And we are using what we call a Mosaic Approach to develop a vaccine that could be one vaccine that could be used all over the world. And the approach uses a viral vector. There's nothing on the market right now, vaccines with viral vectors, but several are being developed. And we basically take an adenovirus, 26, I'll call it Ad26, which can cause a common cold, mild. We make it, genetic engineering is used a lot, so that it doesn't reproduce once it's in your body. But we insert genetic engineering again into its genetic material. These HIV genes, so when the adenovirus is injected into the body, the adenovirus produces these HIV genes that the body sees as foreign. We call them immunogens or antigens, and the immune system mounts a response towards that foreign body and attacks it to get rid of it. And hopefully, if you're exposed again, say to HIV, that same immune response would come back or would still be there and attack it so you wouldn't really get the infection. And what Mosaic means is that we don't use a natural gene, because that natural gene would be for a particular subtype or clade. But Betty Korber at Los Alamos Labs took genes from several different clades or subtypes, and she, like making a mosaic, took pieces from each one so that the gene she created, we call mosaic, when it produces that antigen or immunogen, the body mounts a response, an attack response, if you will, against multiple clades or subtypes. So one vaccine should work for all of them.
I remember way back in the day, she was looking at the challenge with the trying to find the same issue, and she was trying to go way back to the original virus, which that was another concept that they were looking at. Others were looking at trying to find, computerized design for each one. I thought, boy, that would be hard to scale up. So yours seems to be the most practical, because it seems to do, in essence, what they're trying to do, which is work one vaccine against all clades. And most of the clades the diversity is really in Africa, isn't it?
Actually, there's diversity, quite a bit of diversity in Africa, in Asia, and South America. North America and Europe is fairly homogeneously clade B, but the other continents, there are quite a few clades and even mixture of clades that develop. So it's tricky. And I think that the advantage here, one advantage is the practicality, because rolling out vaccine programs on a large scale will be much easier if you just have one and you can just ship batches, millions of doses. And you don't have to, it won't be complicated about this particular kind of vaccine going to this region and this to this region, or to regions where there's a lot of mixture. Then that particular vaccine might not work for everybody.
So one of the things I think Tony Fauci said was that there's kind of criteria by which you must base your development of a vaccine. It has to be scalable and it has to be cost effective and so forth. Do you feel that the direction you're going this will be the case?
Yes, we have already scaled up production a lot. For the vaccine has different components that we can talk about in a minute. We're at final scale or commercial scale already for part of it, and we're well on our way for the rest of it. So we do think it's scalable.
There's multiple steps in the development of the,
Yes,
of the particular kind.
and it's very, very complicated, the production of a vaccine. In most cases, it's many magnitudes more complicated than producing a pill or a drug, especially when you're using live viruses.
Right. So is it sustainable? Does it have a shelf life, or do you? I mean, maybe you're not that far along, but.
Well, we continue to work on that. You have to produce it, and then you have to wait and see what the potency is after a certain amount of time. We will do shelf life studies, or studies that are commonly called end of expiry studies. But we would like to launch the product with shelf life of minimum, well, a minimum of 2 years.
Okay.
But we have to work towards proving that.
Right.
And that's part of the program.
So how do you? So this is the candidate you're moving forward with. You really feel pretty sure of this one above any? Do you have other backup candidates, or?
We are looking at some plan Bs, I would say, in discussion. I can tell you a little bit how we came to the decision on this particular candidate or regimen. So the approach study is a study, a phase 1/2b study, a randomized double-blind study in 393 humans, and testing 7 different schedules or regimens, and then 1 placebo arm. And they are various combinations of the adeno 26 Mosaic vaccine, the MVA Mosaic vaccine, which is a pox viral vector that's been used in other vaccines like smallpox, chickenpox, similar in that family, and also a synthetic piece of the envelope protein from the outer coat of HIV. And simultaneously, in parallel, we did a non-human primate study in rhesus monkeys, testing the same regimens. The difference is in humans, where you can test the safety, the tolerability, and the immune responses. In the monkeys, you can also challenge them with simian HIV and see which regimen protects the best. And then you can look at the immune responses and correlate the immune responses in that particular regimen and try to determine what are the immune markers of protection. Because the immune system's so complicated,
Right.
and nobody knows what is protective.
But you have great correlation in this.
You're right. And so what we found is that the regimen that consisted of receiving 4 injections of the adenovirus at the beginning, at month 3, month 6, and month 12, with that synthetic piece of the envelope protein, GP140, at the 3rd and 4th injections at week 24 and week 48, that in the monkeys, it provided a 94% decreased risk of acquiring HIV when challenged intrarectally per exposure. But what's equally exciting, that after the 6th intrarectal exposure with the simian HIV, 67% of the monkeys who had received that regimen were still uninfected. And that's the first time we've seen data like that in any other vaccine candidate being tested. We found that the immune responses that seemed to correlate with protection were antibodies towards that envelope protein from the outer coat of HIV, and as well as cellular responses towards that envelope. So from the approach study in humans, we found almost exactly the same results. It was that arm that had the best immunogenicity that correlated very closely the pattern of responses to that regimen that was given to monkeys. And what I presented, that data up to 4 weeks after the 4th vaccination, which was at week 52, that was presented from Approach, and the data from the non-human primate study called NHP 1319 was published in the Lancet, a manuscript, earlier this month. What I presented was the follow-up data 1 year after vaccination. And what we saw was that 100% of the subjects who had received that regimen still had the antibody or humoral responses after a year after vaccination, and a high percentage of the subjects still had cellular responses 1 year later. So that's very encouraging that we have persistence or durability of immune responses 1 year after vaccination. I'll also mention that we have demonstrated cross-clade responses, which is what we're hoping from the Mosaic approach, meaning that our antibody responses are not just against clade C, but we have very good responses against a clade B, a clade A. We tested 9 different envelope antigens, and the vaccine produces good responses or robust responses against the 9 different types or antigens, envelope antigens that we tested, and those remained up to a year after. Now we're going to follow up those responses in a 5-year extension study. And that 5-year extension study will really help us see what happens to these responses, and will help inform us when we may need to give a boost of the vaccine. And the safety and tolerability of all the regimens were good even a year after vaccination.
And we'd be remiss if we didn't cover the partners.
Yes, and we are very fortunate to be in partnership with some of the most impressive and people in the field, and some of the most prestigious organizations, and it's a long list, but Dan Baruch at the Beth Israel Deaconess Hospital has been involved from the beginning. We've had support from NIH, Department of AIDS, from the beginning. We also collaborate with the Military HIV Research Program, IAVI, the Reagan Institute, HVTN, and Bill and Melinda Gates Foundation, of course. So the significance of this study is that these data, because it's not just the approach data, but data from the monkey study and some other data and other considerations, prompted us, Janssen, the Bill and Melinda Gates Foundation, and NIH, as well as HVTN, to collaborate and start the first efficacy program using this vaccine concept, this Mosaic vaccine concept using adeno 26 vectors in 5 sub-Saharan African countries. The study is referred to as Imbokodo, which is a word that refers to women. If you hit a woman, it's like hitting a rock, and that refers to the strength of women and became popularized during the anti-apartheid movement. And that started in November of 2017. We're very excited about that, and I would say we're very optimistic about this approach. We're having ongoing discussions on next steps, but we plan to have the data from the Imbokodo study in 2021.
So did you start this this concept, this thought, this proposal with Dan Brook? Was he the one that started it, or was there others that kind of collaborated in the thought to put this forward?
I believe, and I've only been on the program 6 years, and I think the program has started a bit over 10 years ago, and the work with adeno 26 really was developed at Crucell, which is a Dutch vaccine company that Johnson & Johnson acquired. But Dan Baruch started collaborating early on, and I believe he was instrumental in introducing the Mosaic insert concept, and has contributed greatly on many levels, particularly the preclinical levels or the animal studies.
Is Betty Korber still immersed in this, or has she done her part and moved on?
Betty Korber has been critical in what I consider a genius concept. She's not involved day to day in the program, but we are deeply indebted to her and her work and her genius and her generosity.
She's got some really amazing work behind her.
Yes, yes, she does.
Yeah.
And I had the honor of meeting her last year.
We interviewed her way back, and we had a number of folks that were vaccine ideas, way back, and it was always interesting to hear the concepts being proposed, and it's always nice to see one that's actually getting somewhere, and that's exciting.
One thing I want to add, and I don't want to sound corny, but I would say that vaccine trials in the HIV community are not common. There have only been a small handful of concepts tested. So I think physicians aren't universally comfortable and knowledgeable, nor are participants. I refer to participants rather than patients because we're testing healthy people, individuals. But that it has to be a partnership, and that something that I've learned is humility, and the importance of partnering with the community, often through advocacy groups, which kind of help liaise with the community, and at an early stage, explaining what your plans are, asking for feedback, and doing it in a meaningful way, not a tick box way or a check mark, because it's really has to be a partnership with not only the company, if the academic institutions, the clinics that are running the studies day by day, the participants, but also the entire community. And that's what is going to increase the chances of a trial succeeding.
And when you're always looking at a totally new concept, we have to go into the weeds and figure out what are the unintended consequences.
Absolutely.
And it's going to be huge because it's a whole new world.
Yes. Yes.
But it's a brave new world, and we appreciate you for doing so much in the direction of success. You know, it seems like we're headed that direction.
Thank you. I'm personally very encouraged, and my team, the people I work with, our management is very encouraged and very committed to seeing this through.
Right. And you got good people up and down the line at J&J and Janssen.
Yes.
I appreciate so much you being here. Thank you again.
Thank you. Thank you.
Yeah.